So yes, third thing is that the mucus has a low resistance for slippage so that the particles can slide along the epithelium with great ease, because when we have ingested the food, then it must pass through the gastrointestinal tract or alimentary canal, so that it can pass through the different organs, different compartment, and can be processed accordingly. So for that, this mucus actually provides a service over which this food can slide, or these particles can slide along. Okay. Fourth is mucus causes fecal particles to adhere to one another to form the feces that are expelled during bowel movement. So what happens is when the food is digested, the waste material has been formed and it has been separated from the material, that is the particles that is required. Then it reaches to the last part of the alimentary canal, and from there it must be removed out. So for that, if there is no mucus, then the particles will be scattered around and it would be very difficult for the alimentary tract to excrete them out. So to make that feasible, mucus again plays a very important role. It adheres this waste material, the fecal particles together, so that they can move out of the body feasibly. Okay. So fifth, mucus is strongly resistant to digestion by the gastrointestinal enzyme. The very important function that mucus plays is it prevents the layers of the alimentary canal, the surface of the alimentary canal, from being digested or affected by the enzymes that are being secreted in the alimentary tract to digest the food. Okay, and the glycoproteins of mucus have amphoteric properties, which means that they are capable of buffering small amounts of either acids or alkalis. वो हमारे खाने में हो सकते हैं. Also, mucus often contains moderate quantities of bicarbonate ions, जो कि specifically किस लिए होते हैं, कि neutralize the acid. ठीक है, acid जो है, जब HCl is being secreted in the stomach, then it must be neutralized when the food is moving out from the stomach to the small intestine. So for that, bicarbonate ions plays a very important role. We'll see that further in the lecture. Okay, so the next thing is the salivary glands. Salivary glands, there are three pairs of salivary glands. We have parotid gland, sublingual gland, and submandibular glands. Okay. So the function of the saliva are reflected by its constituents. Mucins serve to lubricate the food, make it easier to swallow, and to keep the mouth moist to facilitate masticatory and speech-related movement. If you guys have observed and if you guys have heard the lectures carefully, then you must remember the point that sir told, that our mouth is constantly wet. It's not like it's dry sometimes, and when it is dry, we feel thirst. So in that situation, we generally drink water to keep the mouth moist again. So the moistness is because of the saliva that is present there. Just a moment. So this is because the saliva that is present there, and this mucin is there in the saliva that serves to lubricate the food, make it easier to swallow, and to keep the mouth moist to facilitate masticatory and speech-related movements. Saliva dissolves compounds in food, which is prerequisite for taste bud stimulation and for dental and oral hygiene. Okay, so saliva has a low NaCl concentration and is hypotonic, making it suitable for rinsing off the taste receptors while eating. Infants need saliva to seal the lips when sucking. Saliva also contains alpha amylase, a very important enzyme which starts the digestion of starches in the mouth only. Okay, it doesn't wait to get into the stomach; it starts there and then only when the food is in the mouth, with the help of alpha amylase. And while immunoglobulin A and lysozymes are part of the immune defense system. Saliva contains alpha amylase, it contains NaCl, it contains immunoglobulin A, that is IgA, an antibody, and lysozyme. So IgA and lysozyme actually serve as a function of the immune defense system. If there is any kind of allergen or any kind of foreign material in the food that is not required by the body and that is not the part of the food, then the IgA and lysozyme will take their action then and there only. Okay. So the high HCO3 concentration is saliva. Saliva में बाइकार्बोनेट आयंस की concentration भी high होती है, जिसकी वजह से जो pH है it is slightly towards basic or towards neutral. ठीक है. So the saliva results in a pH of around 7, which is optimal for alpha amylase catalyzed reaction. Every enzyme has a particular optimal situation at which it works properly, and in case of pH for alpha amylase, it is around pH 7 at which it starts working or it works properly. Okay, so swallowed saliva is also important for buffering the acidic gastric juices refluxed into the esophagus. Saliva here performs other function. Another function is it buffers the acid. If you guys have noticed and if you guys remember, then sometimes there is some acidic sensation, some burning sensation in the esophagus due to some acid reflux. Okay, so that must be what, toned down, or that must be buffered by something, and for that saliva plays a very important role. Otherwise, it will affect the cells of your esophagus. Okay, so the secretion of profuse amount of saliva before vomiting also prevents gastric acids from damaging the enamel of the teeth. Because it is an acid, it can affect the enamel of the teeth. Enamel is the outer white covering of the teeth. So to prevent that, saliva also plays a very important role just before we are about to vomit. Okay. So saliva secretion is very dependent on the body water content. A low content results in decreased saliva secretion. The mouth and throat becomes dry, thereby evoking the sensation of thirst. Okay, this is an important mechanism for maintaining the fluid balance. It is all a mechanism, a buffer mechanism developed by the body to maintain different fluids, concentration, their composition, and all. So the secretion rate of saliva is, the rate of saliva secretion varies from zero point 0.1 to 4 ml per minute. It is per minute we are talking about, depending upon the degree of stimulation. Saliva secretion occurs in two steps. एक primary saliva होता है, एक secondary saliva होता है, ठीक है? So the acini and pieces produce primary saliva. This is, if you can see, these are the acini is the plural term. Acinus is the uh, sorry, I said just. So yes, these are the acini, the cells of the salivary gland, and what they do is produce primary saliva, which has an electrolyte composition similar to that of plasma. Primary saliva secretion in the acini cells is the result of transcellular chloride transport. Chloride is actively taken up into the cells from the blood by means of NKCC, sodium potassium chloride co-transporter. ठीक है, if you guys remember, we have studied this in kidney when we were studying about the countercurrent mechanism and all of the functioning of the nephrons. So, and is released into the lumens via anion channels, together which bicarbonate ions are also released there to maintain the balance, resulting in a lumen negative trans epithelial potential that drives the sodium paracellularly into the lumen, and water also follows passively. So basically it is just maintaining the composition of saliva, and it is a mechanism to draw water passively into the lumen of this salivary glands, and where the composition of this saliva is maintained, the primary saliva, and it has an osmolarity of around 300. Okay, later on when the saliva is further processed and it forms the secondary saliva, which is actually released in the mouth, the osmolarity of that changes, it comes to around 100. Okay, so primary saliva is modified in excretory ducts, yielding secondary saliva. As the saliva passes through the excretory ducts, these are the excretory ducts, basically the ducts of the gland through which it passes and comes into the mouth. The sodium and chloride are reabsorbed, potassium and bicarbonate secreted into the lumen. The saliva becomes hypotonic, far below than 100 milliosmoles per kg, because sodium chloride reabsorption is greater than potassium and bicarbonate secretion, and the ducts are relatively impermeable to water. So if the secretion rate rises to values much higher than 100 microliter per minute per gram of the tissue, these processes lag behind. The composition of secondary saliva becomes similar to that of primary saliva. So this concentration must be maintained. Stimulation कैसे होगा? Hypothalamus is involved, salivary center is involved, sympathetic and parasympathetic activation is involved. So in sympathetic and parasympathetic, it works the same way it has been working in all the organs we have studied in. The acetylcholine, the epinephrine, norepinephrine all plays their role according to what have been assigned to them. So what happens is the reflex stimulation of saliva secretion occurs in large salivary glands. Salivary stimuli include the smell and taste of the food, tactile stimulation of buccal mucosa, mastication, and nausea. So basically, saliva secretion is stimulated by the sympathetic and parasympathetic nervous system both together. They together work to secrete the saliva, and for this, the this stimulation comes from hypothalamus to the salivary centers, and the stimuli to hypothalamus reaches through smell, taste, touch, mastication, nausea, etc. Okay, depending upon what the situation is. So sympathetic and parasympathetic nervous system then further initiate the secretion of saliva, and it also makes the parotid glands active for the assigned function. So norepinephrine, actually what it does is it triggers the secretion of highly viscous saliva with a high concentration of mucin via beta 2 adrenoreceptors and CAMP. PIP also increases the CAMP concentration of acinar cells. Okay. So this is all parasympathetic and sympathetic nervous system that comes into action for this secretion. Acetylcholine क्या करता है with the aid of M1 cholinereceptors. So here we have two receptors, one M1 and M3. Okay, so M1 receptors क्या करते हैं with the help of IP3. Acetylcholine mediates an increase in the cytosolic calcium concentration of the acinar cells. So acinar cells में क्या करेगा, calcium concentration को increase कर देगा, under the effect of which neurotransmitter acetylcholine. M1 receptor will do that. So acetylcholine will itself not go inside the cell. It will send a secondary messenger, that is IP3 in the cell, and it will increase the calcium ion concentration in the acinar cells. This in turn increases the conductivity of luminal anion channels, resulting in production of watery saliva and increased exocytosis of salivary enzymes. ठीक है? 2 minutes. What is happening here? ठीक है जी. So yeah, what we were doing is 1 minute. So क्या होगा कि increase the conductivity of luminal anion channel, resulting in production of watery saliva and increased exocytosis of the salivary enzyme. तो कैल्शियम आयन दो फंक्शन करेगा, एक तो वाटरी सलाइवा की प्रोडक्शन शुरू करा देगा, उसका एक्सोसाइटोसिस शुरू कराएगा इन द ल्यूमेन ऑफ द एसिनर सेल्स. उसके बाद क्या करेगा कि ये सलाइवरी एंजाइम्स का भी एक्सोसाइटोसिस शुरू करा देगा. और फिर M3 रिसेप्टर यहाँ क्या काम करेगा, विद द aid of M3 cholinergic receptor, acetylcholine mediates the contraction of myoepithelial cells around the acini, leading to the emptying of acini. जितना भी इनके अंदर जो watery saliva जो stored है, that all will be emptied into the lumen, and then it will pass through the excretory duct and later into the mouth. ठीक है, तो ACH enhances the production of calciferins which cleave bradykinin from the plasma kinkinogen, another compound that is being added to the saliva. So basically it is acetylcholine which is making the release of saliva. Okay. ठीक है जी. Next thing is now the saliva has been released, the food has passed through the esophagus into the stomach. Now stomach has some divisions to make the studies easier. This is the gastroesophageal junction, and it has a sphincter also here, esophageal sphincter. Then this upper part is a cardiac part of the stomach. This is the body of the stomach, and this lower part near the duodenum is the antrum part of the stomach. So this cardia and body together make the fundus part of the stomach, and the lower part is again the antrum part of the stomach. Okay. Then there is this pyloric sphincter through which it is guarding the opening of stomach into the duodenum. So it is pyloric sphincter, it is esophageal sphincter. ठीक है. ठीक है. So we have circular muscle layer, there are longitudinal muscle layers, and also a special oblique muscle layer in the stomach. apart from that, the arrangement is just the same as it has been described earlier for the other organs. Okay, and the gastric pits. Okay, the stomach has the special feature of having the gastric pits in them. Gastric pits are actually very special modification in the stomach. What they do is this: the walls of these gastric pit are lined by different kind of cells which secrete different secretions, which help in the digestion of the food. Okay, so we'll see further what are these things. This is again the different types of the cell. Okay, so the part where this gastric pits are moving inside, they are forming a villi kind of structure, is the straight part is called the stem cell zone, and the last part where it is curved, it is the chief cell zone. Okay, what all of these cells are doing, we'll see further in the lecture. What are they secreting? So this is actually the structure of the gastric pit. Okay, so let's see what are the functions. So the parietal cells, they have parietal cells, they have chief cells, they have different types of cells. We'll see further. So first of all, what are the characteristics of the gastric secretions? In addition to mucus secreting cells that line the entire surface of the stomach mucosa has two important type of tubular glands: the oxyntic glands, also called as the gastric glands, and the pyloric glands. ठीक है? तो ऑक्जेंटिक ग्लैंड्स क्या करेंगे? एसिड फॉर्मिंग ग्लैंड्स, जिसको हम पैराइटल सेल्स भी बोलते हैं, दे सिक्रीट HCl. ठीक है, HCl सिक्रीट करेंगी, इंट्रिंसिक फैक्टर सिक्रीट करेंगी, पेप्सिनोजन सिक्रीट करेंगी और म्यूकस सिक्रीट करेंगी. Pyloric glands क्या करेंगी? They secrete mainly mucus for the production of pyloric mucosa from the stomach acid. They also secrete hormone, which gastrin. Okay, so oxyntic glands कह लो, parietal cells कह लो, they secrete what? Hydrochloric acid, pepsinogen, intrinsic factor and mucus. ठीक है, जबकि जो pyloric glands हैं, what will they do? They will secrete what? They will secrete this gastrin. Gastrin is a peptide hormone. Okay, what is its function? We'll see further. So the oxyntic glands are located on the inside surface of the body and the fundus of the stomach. So this is something you should remember. The oxyntic glands are present in the fundus and body part of the stomach. Uh, this is the structure. So this cardia and body part together make the fundus, and if the oxyntic cells are present then and there only. ठीक है, उसके बाद है कि considering the proximal 80% of the stomach and the pyloric glands are located in the antral, the lower portion of the stomach, and the distal 20 part of the stomach. Let's see what are the different types of cells present there. The oxyntic gland, basically uh, these are present in the fundus part. The antral glands are present in the pylorus part, as we have seen right now. So the different types of cell present is the parietal cells. If you can see the amount of parietal cells in the oxyntic gland, there are no parietal cells in the antral glands. Then there are chief cells which are present again in the oxyntic glands. उसके बाद है the progenitor cells. These are actually the stem cells which will replace the damaged cells, which sometimes get replaced when the body is digesting the food. Okay, and the mucous neck cells. Okay, so these are the mucus secreting cells. Again, in the antral part we have what? The G cells, the red ones, these are the G cells. Okay, and these are the blue ones are the ECL cells. Okay, so we'll see further what are the ECL cells, what are the G cells, what does they do. Yes, G cells secrete gastrin. As I told you, the pyloric gland secrete what? A peptide hormone, the gastrin. So in pyloric gland, it is the G cells which secrete this hormone, that is the gastrin. Okay, and the mucus cells secrete the mucus again. So what does the gastric juices contain? The tubular glands of the gastric fundus and corpus secrete 3 to 4 liters of gastric juice each day. Gastric juice that is being secreted nearly 3 to 4 liters in a day. Pepsinogens and lipases are released by the chief cells and HCl and intrinsic factor by the parietal cells. So chief cells क्या कर रही है? Pepsinogen secrete कर रही है, और parietal cells क्या कर रही है? HCl और intrinsic factor को secrete कर रही है. ठीक है. So mucins and bicarbonates are released by the mucous neck cells and other mucous cells on the surface of the gastric mucosa. Pepsins functions as endopeptidases. Endopeptidases means they can cut in between the peptides. ठीक है, इन बिटवीन क्या करेंगी, पूरा लंबा प्रोटीन है हमारे पास, बहुत बड़ा सा प्रोटीन है, उसको ये कहीं से भी इन बिटवीन काट देंगे डिपेंडिंग अपॉन द प्रेफर्ड साइड्स, और उसको क्या बना देंगे, छोटे छोटे पेप्टाइड्स में कन्वर्ट कर देंगे. ठीक है, दे आर स्प्लिट फ्रॉम पेप्सिनोजन. सो पेप्सिनोजन इज एक्चुअली इन इनएक्टिव फॉर्म, इट मस्ट बी एक्टिवेटेड टू फंक्शन, एंड द एक्टिव फॉर्म ऑफ पेप्सिनोजन इज पेप्सिन्स. ठीक है, दे आर एक्सोसाइटोज्ड और secreted from what, chief cells in the glandular and gastric lumen at the pH of 6. ठीक है, acetylcholine is the chief activator of this reaction. ठीक है, गैस्ट्रिक एसिड क्या है, HCl को जनरली गैस्ट्रिक एसिड बोलते हैं, व्हेन दिस एसिड इज बीइंग सिक्रीटेड, तो उस टाइम पे जो पीक पीएच होगी वो दैट इज अराउंड 1.8 to 4, सॉरी 0.8. ठीक है, उसके बाद क्या होगा, जैसे फूड मिक्स होता है ना इस एसिड के साथ, तो pH generally buffers to around 1.8 to 4, which is optimal for most proteins and different enzymes to function. So it must be lower down, otherwise, sorry, it must be increased. Otherwise, the most of the enzymes won't be able to function, and the gastric lining will be destroyed. Okay. So HCl secretion के लिए हमारे पास जो important चीजें हैं, that is the proton potassium ATPase in the luminal membrane of the parietal cells, which drives H positive ions into the glandular lumen in the exchange of potassiums, and potassium taken up in this process circulates back to lumen via luminal potassium channel. So it is basically just used to operate the channel so that the proton ions can enter inside the cell. So for every H ion selected, an HCO3 ion leaves the blood side of the cell and is exchanged for a chloride ion via an anion antiporter. So if you can see, this is the sodium and potassium, sorry, sodium and proton exchanger. It will take what? It will take sodium and our protons inside, and these protons will further be then used by the, sorry, this sorry, so yes, this sodium potassium X, sorry, sodium proton exchanger, it works in accordance with this water splitting, and it is providing the protons. So most of the proteins are forming that, protons are forming that HCl. Some protons are also getting exchanged using the sodium proton exchanger. Also, there is sodium potassium ATPase, which is working to maintain the balance of the sodium. And again, this, there is this anion antiporter, which will release an bicarbonate outside and will take up one chloride inside to make the HCl. Okay, so HCO3 ions are obtained from CO2 plus OH negative reaction catalyzed by carbonic anhydrase, and this results in the intracellular accumulation of chloride ion, which diffuses out of the cell to the lumen via chloride channel, and thus one chloride ion reaches the lumen for each so sorry protons ion secreted, forming the HCl. Okay. So the activation of parietal cell leads to the opening of canaliculi. Canaliculi is the structure, these are the structure, these are the canaliculi, so which extends deep into the cell from the lumen of the gland. The canaliculi are equipped with a brush border that greatly increases the luminal surface area, which is densely packed with membrane bound sodium, sorry, potassium proton ATPase molecules. This permits to increase the secretion of H positive ions from 2 millimole per hour at rest to over 20 millimole per hour during digestion. So if you can see, there is a tenfold increase in the secretion of H positive ions into the lumen, where it can combine with the chloride ion to form the HCl. Okay. So parietal cells ultrastructure, if you can see, this is the ultrastructure of parietal cell, this is the resting stage, this is the stimulated stage, and if you can see this H positive, K positive ATPases, they are present on the vesicles inside the cell. Once they get the signal from acetylcholine, from different kind of signals through which the parietal cells are activated, these Vesicles will go and fuse with the membrane in the luminal side and will make the these sodium, sorry, proton and potassium ATPase get located on the membrane where it will help in the formation of HCl. ठीक है. So parietal cell structure इन्होंने वही एक resting stage का दिखाया हुआ है, एक stimulated stage का दिखाया हुआ है, and if you can see it here also, ये यहाँ पे इतने हमारे पास जो है ब्रश बॉर्डर्स नहीं है, पर यहाँ पे बहुत सारे ब्रश बॉर्डर्स हैं. So this is all because of the activation. ठीक है. ओके. सो नेक्स्ट इज एंटेरोक्रोमोफिन सेल्स, या फिर हम उनको क्या कहते हैं, ईसीएल सेल्स. वो कहाँ थी? वो थी एंट्रल ग्लैंड्स में. ठीक है. So parietal cells operate in close association with another type of cells called as the enterochromaffin like cells या ECL cells, the primary function of which is to secrete what? Histamine. ठीक है. Histamine, it is a very important amino acid here. The ECL cells lie in sorry, it is not histidine, so it is not amino acid. Sorry. It is histamine. The ECL cells lie in the deep recesses of oxyntic glands and therefore release histamine in direct contact with the parietal cells of the gland. Sorry, guys. So yes, if you can see, they haven't shown it here, but yes, ECL cells are present in close connection with the parietal cells. So what they do is they secrete histamine. Histamine directly comes in contact with the parietal cells, which again act as an activator for the secretion of HCl from the parietal cells. Okay? The rate of formation and secretion of HCl by the parietal cells is directly related to the amount of histamine secreted by the ECL cells. This much control this histamine has on the parietal cells. Yeah. So yes, it is a very important part that you must know. Histamine का receptor इन्होंने दिखाया. This is the H2 receptor. What it does is histamine is released from the enterochromaffin cells with the lamina propria and diffuses through the extracellular fluid to the parietal cell. Histamine act as a paracrine to stimulate acid secretions. As I told you, it will go and stimulate the parietal cells to secrete the HCl. Histamine also stimulates the secretion by the hormonal substance gastrin. ठीक है. तो it will also take help of gastrin to make the release of HCl, which is formed almost entirely in the antral portion of the stromal mucosa in response to the proteins in the food being digested. ठीक है. So in addition, the ECL cells also can be stimulated by acetylcholine released from the stomach vagal nerve endings. You must remember acetylcholine is the one which is actually causing the work of activation here till now. Probably also by the hormonal substances secreted by the enteric nerve system of the stomach. Mostly it is of acetylcholine, but yeah, some role can be played by the hormonal systems also. ठीक है. तो this is just to show you कि this is a thick mucus layer and the pH2 gastric juice is not able to penetrate this mucus layer so that the cells can be prevented. Okay. And the HCO3 being secreted is actually used to neutralize this pH in the duodenum. Okay, we'll see further in the lecture. We'll come to that part also. Cheek. So this is the unstirred layer of the mucus and bicarbonate, the one they have shown. The second, the surface epithelial cells secrete mucus bicarbonate, generate prostaglandins. This is the duodenal mucosa now we are talking about. The food has now reached the duodenum. तो वहां पे क्या होगा, अब पीएच को जल्दी से जो है एसिडिक से इनको एल्कलाइन बनाना होगा. फॉर दैट म्यूकस बाइकार्बोनेट आयंस, sorry, prostaglandins and heat shock proteins, many other proteins come into action to make this pH alkaline. Alkaline tide जो है एक phenomena होता है यहाँ पे. This is because of this bicarbonate ions that are being secreted into the all the kind that is coming inside the duodenum. ठीक है. So yes, this gastric mucosa microcirculation through capillaries is maintained by the continuous generation of prostaglandins, NO and hydrogen sulfide that protect endothelial cells from injury and prevent platelet and leukocyte aggregation. So there are many factors that are involved and the hormonal regulation is done here by the what, gastrin CCK. ठीक है, gastrin and cholecystokinin and another hormone which functions to maintain all of the hormonal regulation. ठीक है, so yes, this is what it happens. So what is alkaline tide? During hydrochloride acid secretion in the stomach, the gastric parietal cells extract chloride anions, carbon dioxide and water and sodium cations from the blood plasma, and in turn releases bicarbonate back into the blood plasma, which in turn release which after forming it from carbon dioxide and water constituents. This is to maintain the plasma's electrical balance, and the chloride anions have been extracted. So the bicarbonate content causes the venous blood leaving the stomach to be more alkaline, the arterial blood delivered to it. Okay, so it is basically the venous blood that is getting more what uh, alkaline. Okay, एक minute guys. Someone wanted to ask something about. So this is what alkaline tide is. Stimulation of acid secretion by gastrin. Gastrin itself is a hormone which is secreted by what, the G cells. As I told you, these are the G cells. There are G cells in the which part, antral part of the antrum part of the stomach, and these G cells actually secrete what, a hormone named as gastrin. These cells are located in the pylori glands or antral glands in the distal end of the stomach. Gastrin is a large polypeptide secreted in two forms. A large form called G34, which contains 34 amino acid, and a smaller form called as the G17, which contains 17 amino acids. Although both of these are important, the smaller one is more abundant. ठीक है, तो ये होता क्या है कि basically gastrin cells in the pylori glands to cause release of gastrin into the circulation, which is then carried to the fundus part of the stomach. The histamine then acts quickly to stimulate the parietal cells to secrete what, HCl. ठीक है, जैसे ही प्रोटीन कंटेनिंग फूड जो है एंड ऑफ स्टमक में पहुंचता है, तो सम ऑफ द प्रोटीन फ्रॉम दीज फूड्स हैव अ स्पेशल स्टिमुलेटरी इफेक्ट, जिसकी वजह से गैस्ट्रिन सिक्रीशन होता है. गैस्ट्रिन जो है हिस्टामिन की सिक्रीशन कराता है, और हिस्टामिन एंड गैस्ट्रिन टुगेदर विल वर्क ऑन पैरेटल सेल्स फॉर द HCl सिक्रीशन. ठीक, so this is the regulation. This is what we have discussed so far. And the vagus nerve, the mechanical stimulus is because of the food, the chewing of the food, and other things. The chemical stimulus is the lowering of the pH because of the secretion of HCl. Gastrin, again a hormone which acts on the parietal cells for the secretion of HCl. Another thing is the acetylcholine from the parasympathetic nervous system, which acts on its uh, designated receptor, the M1 M3 receptors, and will cause the secretion of HCl. Okay, so there are the control at many levels. Okay, there are some kind of the D cells also which secrete somatostatin secreting, which are also called as the somatostatin secreting cell. This is an inhibiting hormone which inhibits the HCl to secrete as histamine. Basically, this is a mechanism working to stop the secretion of HCl so that the pH can be again brought back to what it was in the stomach, as the food has passed down from stomach to the small intestine. Okay, so this is all that mechanism that they have discussed here. Mm. Okay, so gastric acid secretion is stimulated again. This is just a recap so that you guys can get this message clear, that it is stimulated by food intake, deficiency of glucose in the CSF, that is the cerebrospinal fluid, conditioned stimuli over optic, gustatory, and olfactory nerves. This is because when you smell the food and when you see the tasty food, in that case, this conditioned stimuli is also there for the secretion of gastric acid. Acetylcholine directly activates the parietal cells in the fundus, that is through M3 receptors. M1 and M3 receptors both work in that way. Gastrins acting via CCKB receptors on the parietal cells, and the glands in the fundus contain histamine, which are activated by the gastrin and are acetylcholine or beta adrenergic receptors. ठीक है. So the gastric acid secretion is inhibited by somatostatin. It acts via specific receptors on the G cell to inhibit the release of gastrin and thus bring about inhibition of gastric acid secretion. So this is all a intrinsic mechanism in which one molecule is secreting, helping the secretion of other, and when the function is done, then the same molecule is actually helping in inhibiting the secretion also. Okay. So this H2 reception antagonists cimetidine are a class of drug used to block the action of histamine on parietal cells in the stomach, decreasing the production of acid by these cells. These H2 antagonists are used in the treatment of what? Dyspepsia, इंजेक्शन और हार्ट बर्न्स. ठीक है, तो उस केस में क्या करते हैं, उनको एक H2RA ब्लॉकर दे देते हैं, एंड ऑल्दो दे आर सॉरी H2RA दे देते हैं उनको, जो ब्लॉक करता है किसको, हिस्टामिन रिसेप्टर्स को. ठीक है. Although they have been surpassed in popularity by the more effective proton pump inhibitors, which are also called as the PPIs, are a group of drug whose main action is pronounced and long-lasting reduction of gastric acid production. They are the most potent inhibitors of acid secretions available. These drugs are among the most widely sold drugs in the world and are generally considered effective. Okay? So, one drug is omeprazole. Okay? Is a proton pump inhibitor used in the treatment of dyspepsia, persistent or recurrent pain or discomfort in the upper abdomen, peptic ulcer disease, gastroesophageal reflux disease. So these are the different types of disease for which the omeprazole is used. It is a proton pump inhibitor which acts by irreversibly blocking the hydrogen potassium ATP enzyme system, or this pump, or more commonly the gastric proton pump of the gastric parietal cells. The proton pump in the terminal stage in gastric acid secretion, being directly responsible for secretion of what? Proton ions. These proton ions will further then lead to the production of HCl, which can cause increment in the disease. So to prevent that to happen, this omeprazole is an effective treatment. It will bind to this H positive K positive ATPase and will block its function. Then again it will bind irreversibly to this. So with these PPIs are actually given in an inactive form, which is neutrally charged, that is lipophilic, and readily crosses the cell membrane into the intercellular compartments with acidic environments. In an acidic environment, the inactive drug is protonated. So basically जब ये drug हम देते हैं तब it is inactive. When it enters the parietal cell where it gets in acidic environment, उसकी वजह से it gets protonated and gets activated, and activation leads to the what? Blocking of H positive and K positive ATPase, जिसकी वजह से H positive ions जो हैं they want to be released into the lumen gastric gland. Lumen, अगर gastric gland के lumen में H positive पहुंचेंगे नहीं तो HCl बनेगा ही नहीं and the problem will be solved. Okay, so this is how the proton pump inhibitors work. This is the parietal cell structure. Hydrochloric acid intrinsic factor is what they secrete. Okay. Another role is, since there is a continuous need to replenish RBCs, erythropoietic cells of the bone marrow are among the most rapidly growing and reproducing cells in the body, and the process is dependent upon the nutrition, particularly the two vitamins, that is vitamin B12 and folic acid. Okay, both are essential for the synthesis of DNA, particularly for the formation of TTP, thymidine triphosphate. If those two vitamins are not in adequate supply, abnormal RBCs called megalocytes are formed. So why are we discussing it here? Because vitamin B12, as it says, it is important for the maturation of RBC. So it must be reabsorbed while when we intake it into the food. So this reabsorption will be done with the help of what? Intrinsic factor. So the intrinsic factor that was being released by the parietal cell plays an important role in the reabsorption of what? Vitamin B12, which is again very important for the maturation of RBCs. If vitamin B12 deficiency is there, it can lead to what? Megalocytes or disease. ठीक है, तो absorption of B12 कैसे होता है? So vitamin B12 combined with intrinsic factor forming a complex that resist digestion by the GIT enzymes. This complex is absorbed at the terminal ileum by pinocytosis, terminal ileum way, that is near the anus. So vitamin B12 is transported to the liver where it is stored. ठीक है? Pernicious anemia, an autoimmune disease. So this is also an autoimmune disease. A common cause of RBC maturation failure is the failure to absorb vitamin B12 from the GI tract. ठीक है? Intrinsic factor. A glycoprotein is secreted by what? Parietal cells in the stomach. Inability to secrete this factor causes this disease. Intrinsic factor binds tightly with vitamin B12, which helps its reabsorption in the GI tract. And still in the bound state, intrinsic factor binds to specific receptor sites on the brush border membranes of what? Mucosal cells in the ileum. Then the vitamin B12 is transported into the blood by the process of what? Pinocytosis. जहाँ से ये फिर liver में जाता है and then further processing happens. Okay. And the secretion and activation of pepsinogen. We have discussed about pepsinogen. It is secreted by what? Chief cells in what? Oxyntic glands, or we can say gastric glands. So what it do is several slightly different types of pepsinogens are secreted by the peptic and mucosal cells of the gastric glands. Even so, all the pepsinogens perform the same function. When pepsinogen is first secreted, it has no digestive activity because it is an inactive enzyme. So it must be activated. ठीक है, तो कैसे एक्टिवेट होगा ये? However, as soon as it comes in contact with HCl, it is activated to form what? Active pepsin. ठीक है, this pepsin, now when it comes in contact with HCl, the pepsinogen gets converted into pepsin, and now this pepsin is what? The active enzyme, the active protease. In this process, the pepsinogen molecule have a molecular weight of about 42.5 kDa, which is split to form a pepsin molecules having a molecular weight of 35 kDa, kilodaltons. ठीक है, या तो 35,000 daltons कह दो या 35 kilodaltons कह दो. ठीक है, so this chief cell secrete this pepsinogen. अच्छा, pepsinogen जब कन्वर्ट होता है पेप्सिन में, जो कि एक एंजाइम है, this is released by chief cells. another peps Another protease in our alimentary canal are chymotrypsin and trypsin, that will be seen when we'll discuss about the small intestine. Pepsin is most efficient in cleaving peptide bonds between hydrophobic and preferably aromatic amino acids. So as I told you earlier also, that every protease has its preferred site where it will cut and it will form the small peptides from a large protein. So for pepsin, it is between a hydrophobic and preferably amino acids such as phenylalanine, tryptophan, and tyrosine. So regulation of secretion. Pepsinogen secretion occurs in response to two types of signals. Stimulation of the peptic cells by acetylcholine released from what? Vagus nerve. Okay? And stimulation of peptic cell secretion in response to acid in the stomach. So this acid activates this pepsinogen and also it helps in the secretion. It acts as a stimulant for the secretion of this pepsinogen. ठीक है. अच्छा, ग्रेलिन क्या होता है? ग्रेलिन इज अगेन another hormone which is being secreted in the stomach. It is called as the hunger hormone. ठीक है, क्या करता है ये? This hormone is a potent orexigenic peptide that stimulates food intake and body weight gain. So basically this hormone is the one which signals our hunger centers in the brain that yes, I am hungry, I need food. So this hormone is what which sends those signals, and then we feel hunger and the food is eaten by us. Okay? So the ghrelin plays an important role in energy homeostasis. Plasma ghrelin levels were elevated in fasting state and returned to basal levels after refeeding. Okay? Next thing is what? The pancreas, a very important gland of alimentary canal and gland which we also discussed when we were discussing about what? Insulin. Yeah. So it is one of the most important glands of our alimentary tract. This pancreas has islets of Langerhans through which this alpha beta cells secrete insulin, glucagon, etc. And another thing this has is the acinar cells. ठीक है? So these SNR cells are present in the pancreas. These SNR cells are the one which secrete the further enzymes that are required
Excuse me,
for.
ma'am.
Yes,
I don't think the slides are moved. Oh, yeah. Okay.
slice has moved.
Yeah, yeah, it moved now.
Okay, perfect. So, as I, where was I? Yes, I was discussing about acinar cells. So these acinar cells has what? Zymogen granules. These zymogen granules are actually the granules which contains enzymes, all of the enzymes which are required in the small intestine for the digestion of food that is coming from the stomach. The primary level of digestion has occurred, has started in the mouth. Then further it continued in the stomach, and now the food has moved from stomach to the small intestine, where the further digestion and reabsorption of the maximum useful particles will take place, and the waste will be then further forwarded to large intestine, and then at last to the last part of ileum, to the anus. Okay, so these SNR cells has what? Zymogen granules, which actually had this enzymes which help in further digestion of the food, and some of the enzymes present in this zymogen are again in the inactive form that will be activated when they'll come in contact with the other enzymes, which will help in the activation of these inactive forms. Okay, so yes, the SNR cells are actually present in the exocrine part of the pancreas, where these SNR cells are present. And the zymogen is also called as the proenzyme. Why it is called as the proenzyme? Because it is an inactive precursor of an enzyme. Okay. The zymogen granules has, as I told you earlier, has the inactive form of the enzyme, which will be further activated by different kind of the enzymes that are already present and other factors. Okay. So the pancreas secretes zymogens partly to prevent the enzyme from digesting proteins in the cell in which they are synthesized. That is why they are kept in an inactive form in the cells. There is this condition when this duct through which this enzyme must be passed to the small intestine is blocked. Then there is this situation when the enzymes will act on the cell in which they are stored. They will start digesting the cell only in which they are stored. So to prevent that, they are kept in an inactive form. Okay. So what are these digestive enzymes? Pancreatic secretion contains multiple enzymes for digesting all of the three major types of food. Either it is protein, carbohydrates, and fats. It also contains large quantity of bicarbonate ion. Why this bicarbonate ion? So that the food that is coming into the stomach from the stomach is actually acidic, has an acidic pH. So to neutralize that, these bicarbonate ions play an important role. And the most important of the pancreatic enzyme for digesting proteins are trypsin and chymotrypsin, and the one which was in stomach is pepsin. ठीक है, and also the carboxy polypeptidase. Peptidase, that means a protease. It will act on what? Peptides. That is the term, that is the term peptidase has been used. Polypeptidase. ठीक है. So by far the most abundant of these is the trypsin. Trypsin is a very important protease. ठीक है. So trypsin and chymotrypsin split whole and partially digested proteins into peptides of various sizes, but do not cause release of the individual amino acid. Amino acid नहीं बना पाते हैं ये, but large proteins जो हैं उनको small small peptides में जरूर ये digest कर देते हैं. However, carboxy polypeptidase does split some peptides into individual amino acid. तो इंडिविजुअल अमाइनो एसिड में कन्वर्ट करने का काम किसका है? कार्बोक्सी पॉलीपेप्टाइडेज. ठीक है. Thus completing digestion of the some proteins all the way to the amino acid state. Then these amino acids will be reabsorbed and then further used in the body according to their requirement. ठीक है. So this is again they have shown this. So when first synthesized in the pancreatic cell, the proteolytic digestive enzymes are inactive form, जैसे कि हम लोगों ने देखा. So इनकी inactive form का नाम क्या है? Trypsinogen for trypsin, chymotrypsinogen for chymotrypsin, and procarboxy polypeptidase for carboxy polypeptidases, which are all inactive enzymatically. They become activated only after they are secreted into the intestinal tract. The trypsinogen is activated by an enzyme called enterokinase, which is secreted by what? Intestinal mucosa. तो ये सारे के सारे एंजाइम्स जो हैं जब इंटेस्टाइनल ट्रैक्ट में आ जाते हैं तो सबसे पहले ट्रिप्सिनोजन जो है उसको ट्रिप्सिन में कन्वर्ट करता है व्हाट? Enterokinase. Enterokinase is an enzyme again which is secreted by intestinal mucosa, and it is secreted in from the stimulation by the chyme which is coming in contact with the mucosa. Also, trypsinogen can be automatically activated by trypsin. Once it gets activated, it will activate with other molecule, and chymotrypsinogen is activated by trypsin to form the chymotrypsin, and procarboxy polypeptidase is activated in the similar manner. So, enterokinase activates what? Trypsin. Then this trypsin activates chymotrypsin and carboxy polypeptidase. ठीक है? As I told you, कि if these ducts through which the enzymes are entering into the intestinal tract gets blocked, it can lead to serious problem, and the problem is called as pancreatitis. ठीक है? Or inflammation of the pancreas, which occurs when activated pancreatic proteases digest pancreatic tissue itself. ठीक है? So in that situation, it causes what? Pancreatitis, and this can happen because of the gallstones. These stones are formed by various reasons, the deposition of the various kind of salts. ठीक है? कार्बोहाइड्रेट डाइजेशन के लिए हमारे पास क्या है? वी हैव पैनक्रियाटिक अमाइलेज. एक अमाइलेज था हमारे पास अल्फा अमाइलेज इन द सलाइवा, and another amylase is the pancreatic amylase, which hydrolyzes starch, glycogen, and most other carbohydrate except cellulose. You guys must know this thing that our body can't digest cellulose because we don't have an enzyme which is important for the digestion of cellulose, that is cellulase. We don't have that in ourselves, that is why we can't digest cellulose. Okay, so to form mostly disaccharides and a few trisaccharides. Lipid digestion के लिए we have lipases. The pancreatic lipases, we had the gastric lipases also, which is capable of hydrolyzing neutral fats into the fatty acids and monoglycerides. Cholesterol and traces are also there, which causes hydrolysis of cholesterol esterase. Cholesterol esterases are there for cholesterol esters, and phospholipase for phospholipids. Okay. So this is just all what we have discussed so far, the exocrine part of the pancreas and the secretion of bicarbonate ions, as we have discussed earlier, it was there, and again this is what they have discussed. Okay, another thing is the cystic fibrosis transmembrane conductance regulator. CFTR is an ion channel that transports chloride ions across the epithelial cell membrane. Epithelial cell membrane of what? Pancreatic duct cells. ठीक है? So the mutation of this gene causes abnormal transport of chloride ions, and it is a life-threatening genetic disease in which a defective gene for CFTR causes the body to form unusually thick mucus that can result in serious respiratory and gastrointestinal manifestations. Mutation in this single gene are enough to cause CF. ठीक है? Although it is thought that disease severity is linked to the several other genes and proteins, but yes, one and the foremost reason for this disease is the mutation in the CFTR gene, which leads to the thickening of the mucus. Okay, so it is very important. This is again what we have discussed so far. The all of the enzymes, the secretion, and their work. Again, as I was discussing, the acinar cells. So acinar cells are acted upon by secretin and enzyme again, acetylcholine and CCK. So what they do is they CCK will help in discharge of the calcium stores inside the cell. Secretin, acetylcholine and CCK will help in discharge of the calcium stores inside the cell, and secretin will work by the help of a secondary messenger, the CAMP, which will lead to the the secretion of the enzymes, the zymogen vesicles outside the cell, which will then further release the enzymes into the intestinal tract where they will perform their function. So these are the three stimulus: secretin, acetylcholine, and cholecystokinin CCK, which actually works in the secretion of these zymogen vesicles. Next thing is the liver metabolism. A brief summary of liver functions is it helps in the packaging of fatty acids to be stored and transported. It synthesizes the plasma proteins. It forms the known essential amino acid which are required for further functions. It converts the ammonia from deamination to urea, basically the detoxification part. Stores glucose as glycogen and regulates blood glucose homeostasis. Stores vitamins, conserves ions, degrades hormone, and detoxifies the substance. So it is the largest gland in the body and also a very important gland for the body. If you can see, this is the structure of this liver, and it has supply of both oxygenated and deoxygenated blood. Oxygenated blood is to supply the nutrients required by the liver, and deoxygenated blood is to make the hepatic portal system through which it takes up all the toxins that the deoxygenated blood has collected from the body, so that it can be detoxified here. And then further, the detoxified deoxygenated blood is further sent to the lungs, where it get oxygenated, and again the cycle goes on. Okay, so fatty acid foods are absorbed and transported to liver by the lacteal system. This is present in the villi of the small intestine, where it absorbs the fats through this lacteal system, and then it gets transported to the liver. Okay, hepatic portal system, as I was telling you. And this hepatic system, or we can say this liver, is formed of these hexagonal cells. The structure of this hexagonal cell has a central vein, and at its six ends it has a triad of the vessels. ठीक है, तो ये वेसल्स कौन कौन सी हैं? एक बाइल डक्ट है, एक पोर्टल वेन्यूल है, एक पोर्टल आर्टीरियोल है. पोर्टल आर्टीरियोल टू प्रोवाइड इट विद द सब्सटेंसेस रिक्वायर्ड, portal venules so that it can take up the toxic substances it has gathered from the body to detoxify them, and bile duct for the secretion of the bile juices. ठीक है, so it has a triad at every corner of this hexagonal cell, and there are thousands, there are millions of cells that makes this hepatic system, these hexagonal cells. ठीक है, so this was what that was discussed in the last lecture. Now we'll move forward toward the assignment, and I hope no one faced any issue while solving this. Still, if someone has faced the issue, we can discuss it right here. So yes, everyone, they can tell me. Let's start while discussing this question. So this question says, find the correct sequence. What is this thing which is marked as 1? I hope you guys will answer. That will help me and you guys both. So what is this thing which is marked as 1?
Spinster.
It is pyloric sphincter, yes. The thing which is marked as 2? Very easy. It is stomach.
Stomach.
Yeah, right. The thing which is marked as 3 is
Rugae.
rugae. Actually, these rugaes are actually for the absorption and to increase the surface area. Okay. The thing which is marked as 4th is, it is esophagus. Okay.
Esophagus.
Yes. And the thing which is marked as 5th is
Esophagus spinster.
perfect. Perfect. So you were able to solve the question. Next question is, so find the correct sequence. The thing which is marked as 1 is cells which secrete HCl. What are those cells?
Parietal cells.
Parietal cells. Okay. And the parietal cells secrete another thing which is called as, parietal cells और क्या secrete करती हैं HCl के साथ साथ? The intrinsic factor.
G cells.
Intrinsic factor. Intrinsic factor. Okay.
Intrinsic
Uh,
factor.
yes. And the gastrin is secreted by which cells?
G cells.
G cells. Perfect. And the pepsinogen is secreted by
Chief cells.
chief cells. Perfect. Perfect. Okay. What does this diagram indicate? What is this thing which is marked as 1? So 1, 2, and 4 are both the activation signal for the parietal cell. And what are these signals? Acetylcholine, gastrin, and histamine.
And histamine.
Yes. So these are the signals which acts as a activation signal for parietal signal, and the inhibitory signal is somatostatin.
So much standing.
Yes. ठीक है. Next thing is the control of pancreatic juice secretion. In this, the thing which is marked as 1 is
CCK.
CCK. Perfect. The thing which is marked as 2 is
Secretin.
secretin. Perfect. The thing which is marked as 3 is
Food.
food. Actually, food is also acting as a stimulus here. ठीक है. And the thing which is marked as 4 is which organ is that?
Duodenum.
Duodenum. Yes, it is duodenum. Okay, next question. So the thing which is marked as 1 is what is that? Again, the
Duodenum.
duodenum. Yes, it is the duodenum. The thing which is marked as 2 is which duct? Which connects gallbladder to
Cystic
pancreas?
duct.
Cystic duct. Perfect. The thing which is marked as 3 is
Gallbladder.
gallbladder. Perfect. And the thing which is marked as 4 is
Pancreas.
perfect. It is pancreas. So let's move towards some more questions that I have brought for you guys. So, yes, which cell in the stomach secretes hydrochloric acid? We have been discussing it so far and we have discussed it so many times. So, I hope you guys will give a right answer. So, tell me,
Parietal cells.
parietal cells. Perfect. So, answer is B, parietal cells. Okay. The question number 2. What is the main function of pepsin?
Digestion of proteins.
Digestion of protein. Perfect. Which hormone stimulates the secretion of gastric acid? Gastric acid कौन
Gastrin.
सा है? HCl और HCl हम अभी पढ़ के आए हैं. Which hormone is that? Gastrin is that. ठीक है. Question number 4. Which gastric cells produce mucus to protect the stomach lining? Answer is very simple. Which cells are those?
Mucus cells.
Mucus cells. Perfect. What is the function of intrinsic factor secreted by the stomach? It is that
Absorption of vitamin B12.
absorption of vitamin B12. Perfect. Which hormone inhibits gastric acid secretion? कौन सा हार्मोन होता है ये आप लोगों ने
Secretin.
लेक्चर्स में पढ़ा होगा. Yes, it's secretin.
BB.
Perfect. Okay, so next question is which neurotransmitter is responsible for stimulating gastric acid secretion? अब यहाँ पे न्यूरोट्रांसमीटर आ गया जिसकी वजह से गैस्ट्रिक एसिड बनता है. It is what?
Acetylcholine.
Acetylcholine. Perfect. So which enzyme helps in the digestion of carbohydrates in the small intestine?
Amylase.
Amylase. Perfect. So next question is which hormone stimulates the secretion of bicarbonate from pancreas? कौन सा होगा वो?
Secretin.
Secretin again. Yes. ठीक है. So it is again secretin. Next question. Okay, this is the last slide of question we'll be discussing. Which part of pancreas secretes digestive enzymes? It is question number 10.
Exocrine
Exocrine
pancreas.
pancreas. We've discussed this just now that it is exocrine pancreas. Which of the following is a major pancreatic enzyme for protein digestion? कौन सा होता है?
Trypsin.
Trypsin. Which hormone stimulates the secretion of pancreatic enzymes? It is
CCK.
CCK. Perfect. So these were the questions that I have brought. There are still 3 more questions left. You can do it by your own. If you face any problem, you can ask me later in the next lecture also. And apart from that, if you have any confusion, if you have any doubt, you can ask me. I am open for the questions. Any question you guys want to ask?
Currently no. Currently no questions, ma'am.
No questions. Okay, Rajatha. Thank you so much. And if is there anyone in the meeting? No one is in the meeting. So yes, we can conclude the meeting if you don't have any more questions. Okay. Still, if you have
Yes,
some
ma'am. Thank you.
questions, we can ask in the next lecture also. You can ask in the next lecture also. No problem at
Yes, ma'am.
all. Okay then. Bye
Thank
bye,
you, ma'am.
Rajata. Good night.
Bye, ma'am. Good night.